So our next speaker is Chenxuan Pang. I request the IT people to fix the screen in front of us.
It's not working. Technical people in the back. So yeah, maybe you say from where they are.
Yeah. So Chenxuan Pang is from Beijing Normal University, Beijing, China.
It's still here, but yeah, you can. I think you can start. You can start. It's
start.
Just us who
it's
cannot
just
see.
Us, we can
the others can see.
I can see this. I can't see the PowerPoint.
You cannot see as well?
Yes.
Okay. Then
just take a pause.
Yeah. So then the technical people have to help us. Can you see the slides there?
No.
Maybe I can see.
Can see here? I don't know.
Yeah, if you can.
Maybe, so we don't lose.
But
can you move forward? Or?
No.
This is not.
Yeah, it's
not moving.
Maybe I can use this.
Okay.
Okay. Yeah, maybe. Yeah. So you can start while you can try to.
Yes.
Okay. Let's start.
Let's start. Yeah.
Okay. Hello, everyone. My name is Chenxuan Pang, a PhD student from Beijing Normal University. I'm very excited to present our work. Brain age reveals heterogeneous lifespan development of functional networks in depression. I have nothing to declare. Throughout the lifespan, the human brain undergoes continuous and dynamic changes. To understand how the brain evolves during lifespan development, the concept of brain age has gained traction, and the disparity between chronological age and brain age has been referred to as brain age gap, could reflect a brain showed older or younger than normative expectations. And BAG has been recognized as a biologically grounded biomarker of human brain maturation, as it reflected underlying molecular mechanisms, including genetic encoding and plasma proteomics. Also, it has been associated with physiological health, cognitive performance, and mental disorders. The direction of BAG has been linked to different survival curves and lifestyle factors. For example, individuals with high and low BAG had divergent survival curves between around the age of 73. Also, higher BAG has been found to be associated with risky lifestyle, while lower BAG has been related to protective lifestyle. So, opposite deviations in brain age may carry distinct biological and clinical implications. Previous brain age studies have shown that in major depressive disorder, patients had an overall higher BAG compared to healthy controls. However, these group-level case control studies have largely overlooked the bidirectional nature of BAG and failed to elucidate the underlying mechanisms driving such heterogeneity. So to fill this gap, we used two datasets. Dataset 1 was to generate a group-level network population reference. It was consisted of 146 healthy controls. And dataset 2 was our main exploration dataset. It was collected from DIDA project, and it was consisted of 1,065 healthy controls, 1,105 MDD patients. All of them has been pre-processed. And this is a flowchart of our data analysis. First, we established a brain age prediction model based on functional topography. And then we calculated the brain age gap bag and classifying patients with positive bag and negative bag in two groups. We then evaluate correlations between the absolute bag and topography maps and applied gamuts to delineate bag-related regions' growth trajectories. We then used PRS to examine relationships between topography and 17 Hamilton depression rating scores. Finally, we explored gene expression profiles associated with the bag-related regions from Allen Hillman Brain Atlas and Brain Span datasets. First, we calculated brain age for every participant. Specifically, we used NMF to derive individual feature as the input to the brain age prediction model. To avoid bias, we first generated group-level reference atlas in dataset 1. As using it as an initialization, we derived individual functional topography for every participant in dataset 2 based on their time series. And the value of the topography matrix means that the probability of this voxel belongs to this network. We also use combat to reduce the side effect of the brain feature. Using topography as the input feature, the brain age prediction model achieved a high accuracy in healthy controls. Thus, we applied the model parameter to MDD patients and estimate brain age for every participant. And brain age gap was calculated by subtracting the actual chronological age from the predicted age. And we found that there is a significantly larger bag in MDD patients compared to healthy controls. However, substantial heterogeneity was also found. Among the patients, 632 displayed a positive bag, indicative of an older brain, while the remaining showed a younger bag. A negative bag suggesting a younger brain. So we divided patients in two groups, positive bag and negative bag, and we then examined the correlation between functional topography and bag. And we found that in positive bag patients, positive correlations were observed in the dorsolateral prefrontal cortex and ventrolateral prefrontal cortex for the loading of the ventral attention network, which means that when the VAN extends more to these two kind of regions, the brain age gap in this group will show larger. And the negative correlations were found in typical sensory motor network and dorsal attention network regions, which also shows that when these two networks showed a contraction trend, the brain age gap in this group will be larger. And in negative bag patients, positive correlations were found in the ventral attention network loading of the medial orbital frontal cortex, which means that when VAN extends more to the OFC, the bag in this group will be lower. And the negative correlations were observed in typical visual and sensory motor regions, which means that when these two networks contraction, the bag in this group will be also lower. And lifespan modeling also showed accelerated and delayed trajectories in two groups, respectively. We further explored the clinical characteristics in two groups. We used the tenfold PRS to examine topography and HDRS correlation. And we found that in positive bag patients, topography in high-order systems were mainly associated with mood-related symptoms, while in another group, topography in visual and default mode were mainly linked to insomnia problems, and two groups exhibited distinct symptom-related neural mechanisms. We also explored the gene relevance of bag-related brain maps. We used Allen Hillman Brain Atlas dataset and using Metascape to perform a gene enrichment analysis. And we found that in positive bias patients, the genes were mainly enriched in transporter complex, dendritic tree, and synaptic signaling, while in another group, the genes were mainly enriched in positive synaptic membrane, dendritic tree, and synaptic signaling. Finally, we explore the gene associations during development. We used brain span datasets and selected 4 key neurodevelopmental gene sets. We divided brain tissues into back-related regions and non-back-related regions, and we then fitted the transcriptomic trajectories and calculated the gene expression difference. And we found that in positive bias patients, back-related regions exhibited higher expression for myelination and axon development, but lower expression for dendritic and synapse development, while in another group, this difference pattern was totally opposite. Overall, we classified patients by back direction in 2 groups, and we for the first time revealed heterogeneous patterns of deviation from typical neurodevelopment in MDD. These groups exhibited divergent topographic back associations, neurodevelopmental trajectories, clinical characteristics, and gene expression profiles. Our findings provide new insights into the neurobiological mechanisms of MDD across the lifespan and offer potential insights for personalized diagnosis and treatment. Finally, I would like to thank my supervisor and collaborator. That's all. Thank you for your attention, and we can have a further discussion at poster 1370. Thank you.
Thank you. Thank you. There's a question I would like to ask, which are in the audience response. Are you the person who has asked?
No.
Okay, then. There may exist some regression-related, these are mainly regression effects, so is there some corrections you have applied? What are the, I think the essence of the question is about the multiple corrections that one may want to do.
Maybe you want to ask.
So because there are a lot of regression that you have done, I think is there some corrections that you have done? What are the corrections? Maybe you can elaborate on that.
What?
Statistical correction in terms of the so many comparisons. I think that's the essence of this question.
Yes, the correlations between.
So, for example, I think between the 2 groups, you have looked at the behavioral
Yes.
scores and these topographies, right?
Yes.
But are there any comparisons for the corrections for the multiple comparisons? So you are comparing across different factors.
Oh, yes. Yes, it's a really good question.
Yes.
We have considered it, and I want to do it in future.
Okay.
Yes, thank you.
Okay. And another question is, does BAG have a bigger effect size for case control discrimination than individual brain measures?
Yes, when we divided 2 groups and the effect size is improved, right? Yes.
Okay.
Hi, very interesting talk, thank you. I'm just wondering whether you have controlled the age distribution, the actual age distribution between MDD and HC, because there's one plot in the slides which just shows the actual age distribution. Does not seem to be very similar. I mean, the medium one is about 25, the other one seems to be around 30. And also the outline point of MDD and HC, the actual age distribution seems to be slightly different. I guess a QQ plot would be very helpful to clarify that, right?
Yes, your question is really useful for me, and we had performed a two-sample t-test to compare the age distribution of these 2 groups, and we found no significant difference. Yes.
Okay, sure.
Thank you.
Thanks.
Yeah, thank you very much.
I have a question if there is no other question. So one more question. So it's a very interesting study. One thing that I was wondering, have you looked into specifically this core periphery axis, in terms of the brain age gap? Is there a difference across the core networks and the periphery networks in terms of sensory versus integrative kind of networks of the brain? More in that kind of a sort of separation you have done. Like you have looked at the different areas in terms of the brain age graph with the genetic expressions, but have you looked at the sensory versus the integrative networks specifically?
You mean in other data sets?
Yes, in the same data set. Or
in same data sets.
Yeah.
And
so are you aware of this, the Dan Margulis's kind of approach of looking at gradients of the networks? I was just wondering if you have thought about the analysis in those terms. If it's not, it's okay.
Yes, thank you.
Yeah.
I will do this in future.
Okay,
Thank you.
Thank you.